Hello everyone, welcome back to my channel. In this tutorial, I will show you how to retrieve gene annotation from Ensembl BioMart for enrichment analysis. We will use Aedes aegypti as our example. What is BioMart? BioMart is a query system that allows us to retrieve information from Ensembl databases. To use BioMart, we need to understand three important terms: dataset, attributes, and filters. A dataset specifies the organism and genome assembly that we want to study. Attributes are the columns of information that we want BioMart to return. For example, we can request the Ensembl gene ID, GO term, GO category, and Entree gene. Filters are optional conditions used to restrict the results. For example, we can retrieve annotation for a particular chromosome or only for a specific list of genes. BioMart can be accessed in two ways. The first option is the BioMart web interface. The second option is the BioMart package in the R programming language. This method is scripted and reproducible, which means we can save the commands and run the same analysis again. For a bioinformatics pipeline, I prefer using the BioMart package because it makes the complete analysis easier to reproduce. Why do we need BioMart? For common model organisms such as humans, mice, and fruit flies, ready-made annotation packages are generally available. However, for many non-model organisms, we may not have a commonly installed. In that situation, clusterProfiler cannot automatically recognize our genes. We must provide the relationship between the gene IDs and their functional terms. BioMart helps us create this relationship. First, BioMart returns a table containing the gene ID, GO term, and Entree ID. We then convert the GO annotation into a TERM2GENE table. This is simply a two-column table that connects each GO term with its associated gene. Finally, we provide this table to the enricher function in clusterProfiler. BioMart's main job is to prepare the gene annotation, while clusterProfiler performs the enrichment test. The first practical step is to identify the correct BioMart dataset. We should not guess the dataset name. Dataset names can also change between Ensembl releases. First, we load the BioMart package. Next, we connect to the Ensembl Metazoa BioMart because Aedes aegypti is an insect species. We then use list data sets to retrieve all datasets available on the server. The same method can be used to identify available attributes. After selecting the correct dataset, the next step is to retrieve the required annotation using the getBM function. In this query, we request four attributes. The first attribute is Ensembl_gene_ed. The second attribute is GO_ed. This returns the gene ontology accession, such as GO terms beginning with GO. The third attribute is namespace. This tells us the GO domain associated with each term. The fourth attribute is EntresGene_ed. This provides the NCBI Entree gene ID, which can be used for KEGG enrichment analysis. We retrieve all four attributes in one BioMart query and store the result in an object called BM. We then save the annotation as a tab-separated file. After downloading the BioMart annotation, we reshape it into two lookup tables. The first table is the GO mapping table, called GUF_map. This GO mapping table will be used to create the TERM2GENE input for GO enrichment analysis. The second table is the Entree ID mapping table, called ED_map. This table connects each Ensembl gene ID with its corresponding NCBI Entree gene ID. Before running the analysis, let us discuss four common problems that you may encounter. The first problem is an incorrect dataset name. Always run list data sets before starting the query. The second problem is that column headers can change. The third problem is an unexpected GO domain value. The fourth issue is missing Entree ID. This is normal for a non-model organism and does not necessarily indicate an error in the script. Sometimes the Ensembl genome server may be temporarily unavailable and return an HTTP 500 internal server error. In that situation, we can use an alternative annotation source. One option is to search Annotation Hub for an available ORGDB resource. Another option is to download a gene association file, or GUF file, from Vectorbase or another current annotation provider. So, this is the complete idea behind our BioMart workflow. We first identify the correct dataset, retrieve the required annotation, prepare separate lookup tables for GO and KEGG, and then use these tables in clusterProfiler. Now, let's begin running the R script step by step. Step 1. Install and load the required R packages. First, we will install and load the R packages required for this analysis. The installation commands shown at the top should be run only once. If these packages are already installed on your computer, you do not need to install them again. I have placed these commands inside suppress package startup messages so that unnecessary package loading messages are not displayed in the console. Once all the packages have loaded successfully, we can proceed to find the correct BioMart dataset for our organism. Step 2. Find the correct BioMart dataset. Now, we will identify the correct BioMart dataset for Aedes aegypti. First, we load the BioMart package. Then, the use Ensembl genomes command connects R to the Ensembl Metazoa database, which contains genomic information for insects and other invertebrates. Next, we use list datasets to retrieve all available organism datasets. Because this list is very large, we use the grep command to search for the word Aegypti in the dataset descriptions. Now that we know the correct dataset name, we will connect directly to the Aedes aegypti dataset using use Ensembl genomes. Next, we use the getBM function to retrieve 4 important attributes. The downloaded annotation is stored in an R object called BM. We rename the columns to make them easier to understand and save the complete annotation as a tab-separated file. The getwd command shows the working directory where this file has been saved. In the terminal, I use the ls command to confirm that the file was created. Then, I run head command to display its first 5 lines. As we can see, the file contains the gene stable ID, GO term accession, GO domain, and NCBI gene ID. Now that the annotation file is ready, we can use it to build the GO and KEGG lookup tables. Now, we will read the saved BioMart annotation file and prepare it for GO enrichment analysis. We use read_tsv to import the file into R and store it as an object named BM. We perform 2 simple checks. The nrow command shows that our annotation contains 101,982 rows. This large number is expected because one gene may be connected to several GO terms. Table command counts the records belonging to each GO domain. We can see annotations for biological process, molecular function, and cellular component. Next, we create an object called guf_map. Transmute command selects and renames 3 important columns. Filter command then removes rows with missing GO terms, empty GO terms, or invalid GO aspects. Finally, distinct removes duplicate gene to GO relationships. Next, we will create the id_map table for KEGG enrichment analysis. BioMart provides both the Ensembl gene ID and its corresponding NCBI Entrée gene ID. We select the Ensembl gene ID as gene and the NCBI Entrée gene ID as Entrée. Next, we will retrieve the biological name associated with each GO accession number. We select the term column, which provides the readable name of each GO term. Next, we will verify and correct the GO categories using the GO DB package. First, the annotation DBI select function searches all unique GO accession numbers from a BioMart annotation. Next, we create onto2 aspect to convert these ontology abbreviations into the standard GO aspect codes. *BP becomes P. *MF becomes F. *CC becomes C. We then rebuild the guf_map table using the gene stable ID and GO term accession. Now, we will perform GO enrichment analysis using our significant genes. First, we import the differential expression results from the file. Next, we extract the unique gene IDs from the gene_ID column and store them as SIG_genes. Next, we create a function called run_go. This allows us to use the same enrichment settings for all 3 Go categories. The TERM2NAME argument provides readable Go term names from our term2name_Go table. The initial p-value cutoff is set to 0.05. Multiple testing correction is performed using the Benjamini-Hochberg method, indicated by BH. The q-value cutoff is set to 0.2. We also include only Go terms containing between 5 and 500 annotated genes. Finally, we run the function 3 times. Finally, we convert our significant ensemble gene IDs into entree gene IDs using the id_map lookup table. Now, we will perform KEGG pathway enrichment analysis using the entree gene IDs. First, we use the enrich KEGG function from the clusterProfiler package. Enrichment result is stored in an object named KK. Next, we convert this enrichment object into a regular data frame named KEGG_results. Save the complete results as a CSV file. We also print the results in the R console so that we can examine the enriched pathways. Next, the script checks whether any significant KEGG pathways were identified. If the result table contains one or more pathways, the dot-plot function displays the top 20 enriched pathways. The title of the plot is set to KEGG Pathway Enrichment, Aedes Aegypti. Finally, the plot is saved as a PDF file named kegg.plot.pdf with a width and height of 8 inches. After completing the enrichment analysis, we can visualize the significant results using a dot plot. In the plot shown here, each row represents an enriched pathway. The horizontal axis shows the gene ratio, which represents the proportion of input genes associated with each pathway. These results suggest that the significant genes are strongly associated with detoxification and metabolic processes in Aedes aegypti. Now, we will perform GO enrichment separately for biological process, molecular function, and cellular component. We use the enricher function to analyze biological process terms. The gene argument contains our significant genes, while universe should contain all genes tested during the differential expression analysis. The TERM2GENE table is created from GUF_map. Here, the aspect code P selects only biological process annotations. The TERM2NAME argument adds readable Go term names to the results. We use a p-value cutoff of 0.05 and the Benjamini-Hochberg method to correct for multiple testing. The q-value cutoff is set to 0.20. The MINGS size and max_GS size arguments restrict the analysis to Go terms containing between 5 and 500 annotated genes. The result is stored in the object GO_BP. We then repeat the same analysis for molecular function. Finally, we use the aspect code C for cellular component and save the result as go_cc. After completing all three analyses, we convert each enrichment object into a data frame and save the results as 3 CSV files. Next, we create the Go enrichment .plots. Before creating each plot, the script checks whether the corresponding result table contains at least one enriched Go term. If biological process results are available, the top 20 enriched terms are displayed and the plot is saved as plot.pdf. The conditional if statements are important because they prevent R from trying to create an empty plot when no significant Go terms are identified. After completing the enrichment analysis, we can visualize the significant results using a dot plot. In each dot plot, the vertical axis displays the enriched Go terms. The size of each dot represents the number of significant genes associated with that Go term. The color represents the adjusted p-value, indicating the statistical significance of the enrichment. We have now completed the Go enrichment analysis and saved both the result tables and the visualization files for all three Go categories. Thank you for watching this BioMart tutorial. If you found this video helpful, please like the video and subscribe to the channel. If you have any questions or suggestions, leave a comment below. See you in the next bioinformatics tutorial.